Hi guys, the purpose of this video is I'm going to be going through the genetics problems on the review to help you prepare for your test. So looking at this first problem, we have a woman who is homozygous for freckles, which it says is a dominant trait, and has type AB blood. So first thing I'm going to do is figure out, what is her genotype. We'll use F for freckles, and since she is homozygous, it means she has two of the same allele, and it's the dominant trait, so she is big F, big F. And then she's type AB blood, so her genotype for the blood type is AB, because that's the only thing that AB blood can be. Now we have a man who is heterozygous for type A blood and has no freckles, so we'll do the freckles first. If he has no freckles, his genotype must be little f, and he's heterozygous for type A blood, so he has type A blood, so he has an A. Since he's heterozygous, the other allele must be O, because O is recessive. And if he was big A, then he would be homozygous rather than heterozygous. Okay, so now we have the genotypes of our parents. So we're going to make our Punnett square using every possible combination of those alleles. So for the woman, she could have certain eggs have F, big F, and then A. She could also have eggs that have F and then B. Now, if we wanted to do this full square, half of her eggs are F big A and half of her eggs are F B, so we could do it like this. And if it, helps you to do the whole Punnett square, then go ahead and do that. But since these two columns are exactly the same, and these two columns are exactly the same, it's like how, with ratios, 1/2 is the same as 2/4. I can just cross these columns out. I don't need them, actually. But again, if you want to do them, feel free. Now for the man, some of his sperms will contain little f and A, and some of his sperms will contain little f and O. And same as with the woman, half of his sperms are little f and A, and half of his sperms are little f and O, and since these two columns are the same, or these two rows are the same, and these two rows are the same, I don't actually need those two rows either. So now this has simplified it quite a bit since we had some homozygous traits in there. We'll just go ahead and bring down all the alleles into their appropriate boxes. So big F, little f, big A, big A, big F, little f, A, B, big F, little f, A, O, and big F, little f, B, O. All right, so now to do the genotypes, it looks like we do have 4 different genotypes. So we have 1 big F, little f, A, 2, 1 big F, little f, A, B, 2, 1 big F, little f, A, O, 2, 1 big F, little f, B, O. So those are the genotypes. Now for the phenotypes, these two both have type A blood. So for phenotypes, we have 2 with freckles and type A, 2, 1 with freckles and type AB, 2, 1 with freckles and type BO. So there's both our genotypes and our phenotypes. You could have also expressed these as percentages. So these would be, the genotypes would be 25% each, and then for the phenotypes, you'd have 50% of the type A, 25 for AB, and 25 for BO. For this problem, we're looking at calico cats. So remember that for the cats, the orange and the black coloring are codominant, and they are also sex-linked. So the black male cat, let's start, since we're dealing with a sex-linked trait, we need to use X's and Y's. So the male cat would be XY, and the female cat would be XX. And then the orange and black traits are found on the X chromosome, not the Y chromosome, because it's a sex-linked trait. So the black male cat would have, put a little B on his X, and then since the female cat is orange, she would have O's on both of her X's. So put the female here on the punnet and the male on the side. And then we'll just fill this out. So what we end up as a result is these two are both calico females, so females that have orange and black. So we could say 50% or 1 if we want to do it in ratios. Calico female to 50% or 1. These would be orange males. So only the female cat can have orange and black, because only the female cat has 2 X chromosomes. Polygenic traits are just traits that are controlled by multiple different genes. So not multiple different alleles necessarily, but multiple different genes, probably in different places on different chromosomes. Classic example of polygenic traits in humans is height. There are a lot of different genes that contribute to height. Other polygenic traits include things like skin color or eye color. Now on number 6, we're looking at cows, and we have a white cow and a red cow, and they produce a calf that is spotted with red and white. So since there is both red and white, and they're not blended together to make pink, that would be a codominant trait, where the incomplete dominant would have been if they blended together. But in this case, we get spots of both colors, so that would be a codominant trait. If 2 spotted cows have baby calves together, then the genotypes of the spotted cows are RW, red and white, because neither trait is dominant over the other, so they both get their own letter. We don't use uppercase and lowercase. Those are only used for dominant and recessive traits. For codominant traits, they each get their own letter. So there's one spotted cow and the other spotted cow, and we'll fill this out. And we should see that this box would represent a white calf. So the percent chance of the white calf would be 1 out of 4, 25% chance. So this problem is giving us an example of a double heterozygous cross. So we've got 2 traits. Both of the parents are heterozygous for everything, and these are both simple dominant recessive traits. So I'm not sure if Mr. Kane went over this with you, maybe he did, but the expected ratio for a cross like this is 9 to 3 to 3 to 1, meaning 9 of having both dominant traits, 3 of one dominant and one recessive, 3 of the other recessive, and then the other one dominant, and 1 of having both recessive traits. You can feel free to do the Punnett square, but you should see that for the phenotype ratios, you should come out with this. So now the question is, do these numbers match that ratio or not? If they don't, then we can say that these genes appear to be linked. It appears that they are not assorting independently, like we would expect from our Punnett square. So we're going to do a little bit of math now for the chi-square. Let's go ahead and make our observed and expected, and observed minus expected squared divided by expected columns. I need 4 in here. Let's see. Okay, well, it's a little messy, but it'll do. So our options for plants, we have, I guess, thick stem, curled leaves. We have thin stem and straight leaves. We have thick stem and straight leaf. And we have thin stem and curled leaves. Okay. And then our observed numbers are going to come from here. The numbers we're given in the problem. So 700 of those, 500 of those, 100 of those, and 80 of those. Now for the expected, I expect this ratio up here to have held true for my plants. So my total number of plants, it looks like, is 1,380. So out of that 1,380, I expect that 9 out of 16 would have both dominant traits, 3 out of 16 each would have the mix, and then 1 out of 16 would have both recessive traits. So I'm going to take 9 divided by 16 and then times my 1,380 here, total plants, and I get 776 expected for the thick stems and curled leaves. Thin stems and straight leaves, those are the recessive traits. So I need to take 1 divided by 16 times 1,380, and I expected 86 plants of that type. And then the last 2 are the 2 that have 1 dominant, 1 recessive trait. So I'll take 3 divided by 16 and then times 1,380, and I get, let's see, I'm just going to round it to 259 for each of those. Okay, so I'm going to go ahead and do the math for the last column, and then I'll just put that back up. Okay, so when we add all of our values in the table together, we end up with a chi-square value of, it's all 2's. And you might have gotten a slightly different value if you rounded differently. Now we have 3 degrees of freedom since we have 4 categories, which means the critical value is 7.81. Now clearly our chi-square value is much larger than our critical value. This means we need to reject the null hypothesis, or we reject what our Punnett square would have told us should be the ratios we got, which was this, again up here. That means that most likely these are, in fact, linked genes, not assorting independently. They are close together on the same chromosome, so they're tending to cross over together or stay together during that crossing over process. Okay, so for this one, we've got another 2-trait cross, but this one's a little more complicated because we have a sex-linked trait in there. So we have a colorblind woman who is heterozygous for tongue rolling. So since this is a woman, she will be XX, and she is colorblind. I think I'm going to use B's for colorblind since different sizes of B's are easier to tell apart than different sizes of C's. So since she is colorblind, she's going to have 2 little b's for her alleles. Because colorblind is a sex-linked recessive trait. And then she's heterozygous for tongue rolling, so we'll use T's for the tongue rolling. So we'll do big T, little t. The man, so he is XY, he has normal vision, so he'll have a big B on his X, and again the Y doesn't have the allele on it, so we'll just leave that blank. And then he is also heterozygous for tongue rolling, so big T, little t. Now doing every possible combination of alleles, the woman could be X little b big T, or she could be X little b little t. Those are her only 2 options. We'll put those in. And I'm once again going to cross out these 2 columns because they're superfluous in this case, because those would just be identical copies of those 2 columns. But for the man, we have, he could be X big B big T, he could be X big B little t, he could be Y big T, or he could be Y little t. So there are 4 different possible combinations that his sperms could carry, so we will need all 4 rows. Put those in there. Y big T and Y little t. All right, so I'm going to fill out this square now. So now if we look at the results of this, we are looking for a female child with normal vision who cannot roll her tongue. So this bottom section, those are the male children. Up here we have the female children. Now the female children all have normal vision because they all inherited an X big B from their father. And then cannot roll their tongue would just be this one that has 2 little t's. So the percent chance of a female child with normal vision who can't roll her tongue is 1 out of 8, which is a 12.5% chance. Now for these pedigrees, you're going to want to be able to identify traits based on a pedigree, but for this practice, it was just practicing drawing them to see if you could draw something that would fit that pattern. So autosomal dominant trait means not sex-linked and it's dominant. So there's different things you could do for this pedigree, but let's say both parents or grandparents, I suppose, the first generation had this trait. In that case, let's say potentially they could be heterozygous, right? So we could get a situation where maybe one child has it and the other child here doesn't, because if they were both heterozygous and this was a dominant trait, you could get a child with the recessive trait out of this. Now, things you would be looking for in a dominant trait would be if you have 2 parents who do not have the trait, like these 2 individuals, it would be impossible for either of their children to have that trait. So we would not expect to see this one filled in, for example. That would not be indicative of a dominant autosomal trait. We could also expect to see a situation like over here, if we had one parent who had the trait and one parent who didn't, it would be possible for a child to have the trait. Like so. So this tree I've drawn here could be an example of that sort of trait. Also things to look for would be males and females are about equally affected. You wouldn't see a lot more of one sex having the trait than the other because that would indicate it was a sex-linked trait. Now for the recessive trait, we'd almost sort of could see the opposite sort of thing happening, where we maybe have neither parent of these two parents has the trait, but they could have a child who does have the trait. Because it's a recessive trait, so it's possible for the child to have the parent trait even though the parents didn't. Now, it can vary with pedigrees. Some pedigrees will show you carriers half shaded, some won't, or some will expect you to sort of imply, but we could potentially have shown that these two were carriers of the trait, and then that's how this offspring got the trait. You would never expect to have a situation where, for example, both parents had the trait and the offspring did not, because if this is a recessive trait, if both parents had the trait, then these offspring would also have to have that trait. And again, since this is an autosomal trait, we would expect approximately, like it doesn't have to be exact, but approximately the same number of males and females would have the trait. So for a sex-linked trait, one thing we would expect to see is a much higher rate of males affected by the trait than females. So for example, maybe we could have a situation where this female was a carrier of the trait, and maybe it would show that and maybe it wouldn't. And then we would have her son having the trait, and even though the father was totally normal, and then his daughter would end up being a carrier of that trait. But really this pattern right here, where you have parents who don't have the trait but a female carrier and then a son who does have the trait, that's kind of what you're looking for in a sex-linked recessive trait. You'd also expect to see many more males being affected actually having the trait. So maybe this female inherited that gene from her mother, and then her son has it. So you'd see a lot more males actually having the trait as opposed to females. It'd be uncommon to see the females having the trait. All right, so I hope that helps. Please comment on the assignment if you have other questions or just things you want to go over before the test.